Now we are discussing the factors affecting the affinity of the pigments to the particular gases. Now the very first factor, this is the specific variant of the hemoglobin. So that means that if we are taking up like three vertebrate animals, all the three animals will be having some structural difference in the hemoglobin, as we have discussed earlier. So there can be a difference of the arrangement, it can be a difference in the globin protein. So the specific variant of the hemoglobin, it is very important as per the affinity is concerned. Second thing is cooperativity of the binding in the multimeric forms. So that means that some of the pigments, they have the capability to bind uh, more molecules of the gas. The third, which is very important factor, this is the temperature. So as the temperature it increases, the pigment it tends to release the gas. So if we take the temperature at the lung level, as we are inspiring the air inside our lungs, so we have got a lower temperatures at the lung level. So that helps the pigment to bind to oxygen. As the temperature it increases at the tissue level, it helps the pigments to release the gas. So that means a lower temperature will increase the affinity of the pigment towards oxygen, and a higher temperature will decrease the affinity of the pigment towards the oxygen. Next factor is the pH. So pH is again a very important factor, and we'll be taking out one or two slides on the pH as the major factor affecting the affinity of the pigments. Next is the carbon dioxide. So partial pressure of carbon dioxide is again very important factor for the pigments to take up the oxygen. So as we are more concerned with the oxygen transport, the partial pressure of carbon dioxide, if it is high, then there will be a lower affinity of hemoglobin towards the oxygen. If the partial pressure of carbon dioxide is low, there will be higher affinity of the pigment towards the oxygen. Next factor is the organic phosphates. So various phosphates, the organic phosphates, they may affect the affinity of the pigment. Next are the inorganic ions. So many ions, they can affect the affinity of the pigment towards a particular gas. So these were some of the factors which are affecting the affinity of the pigment. So here the most important the factors are the temperature, the pH, and the partial pressure of the carbon dioxide. So these are the major factors which are affecting the affinity. If we are going for the effect of carbon dioxide, this graph is very clearly showing that if you have a high carbon dioxide partial pressure, then you have a less affinity of the pigment towards the oxygen. Now here in this graph, at the vertical axis, we have got the hemoglobin concentration. So this is the percentage hemoglobin concentration. So that means that should be, I think, we should write here as hemoglobin saturation. So at the vertical axis, you take hemoglobin saturation as percentage. So if we are going up, we are having more of the hemoglobin saturated with oxygen. At the horizontal axis, we have got the oxygen tension. So this is the partial pressure you can see of oxygen. So if we have, if you see, suppose if you're taking the blue curve, so this is the medium CO2 partial pressure curve. As the oxygen tension increases towards the right side, the hemoglobin saturation also increases with oxygen. So if the carbon dioxide is low, see the red colored graph, if carbon dioxide is low, then more of the hemoglobin will be saturated with oxygen. So it goes to left side. And if the carbon dioxide partial pressure is high, then it decreases, or as shown in the green curve, you can very well see that the hemoglobin saturation will decrease as the partial pressure of the carbon dioxide is high. So this graph, it is very clearly showing you the percentage of hemoglobin saturation with the carbon dioxide partial pressure. So this particular table, it shows you the affinity of the hemoglobin under different conditions. Like if we go with the first horizontal line, the upper line, the violet line, this is the gas exchange surface. That means the lung level. So what is oxygen concentration at the lung level? It is high. What is the carbon dioxide concentration at lung level? It is low. So the affinity of the hemoglobin for oxygen, it is high at the lung level. So it is joining the oxygen. So what is the result? It is the oxygen is attached to the pigment because the affinity of the hemoglobin for the oxygen is high at the lung level. So what is the result? It will combine with oxygen, it will take up oxygen and forms the oxyhemoglobin. Now if you see the lower line, it is at the tissue level, the respiring tissues. So what is the oxygen concentration at the tissue level? It is low. What is the carbon dioxide concentration at the tissue level? It is high. So what is the oxygen affinity for the hemoglobin? It is again low. Why it is low? Because at the tissue level we have got higher PCO2. That means we have a high partial pressure of carbon dioxide. So that means the affinity will be low, as we have seen in the previous graph. So see the green graph, if CO2 is high, we have got a low concentration of hemoglobin saturated with oxygen. So that means we have a low affinity of hemoglobin for oxygen at tissue level. So what is the result? Oxygen is released by the pigment. So that means the hemoglobin will release the oxygen. So the oxyhemoglobin will become hemoglobin and oxygen will be released at the tissue level because the affinity is low. Why it is low? Because partial pressure of carbon dioxide is higher at tissue levels. Okay, now this is a particular graph which is showing you the oxygen dissociation curve. Now this is called as ODC, or oxygen differentiation curve, or oxygen dissociation curve, not differentiation. You should write it as oxygen dissociation curve. Now at the vertical axis we have the percentage saturation. So this is the percentage saturation of the hemoglobin. So at the horizontal axis we have got the partial pressure of oxygen. So you can very well see that the first curve is for an elephant and second curve is of the horse, third is for the man, fourth is for the sheep, fifth is for the fox, sixth is for the cat, seventh is for the rat, and eighth is for the mouse. Now in this particular graph, it is seen that at a particular oxygen partial pressure, what what content of the hemoglobin is saturated. Now, if you see the first graph for the elephant, and if you see the partial pressure at 40 mmHg of oxygen partial pressure, the hemoglobin curve is near to 85 for elephant. And if we go for horse at 40 oxygen partial pressure, it is near to 78. So it is lower than the elephant. If you see man at 40 partial pressure, the percentage of hemoglobin saturation is near to 70. If you see the third line, it is near to 70 at 40. And if you go with 80, I'm just comparing the first three animals. If you go for 80 oxygen partial pressure, what is the HB saturation? For elephant, it is near to like 98. For horse, it is again near to 95. For man, it is near to 98. That is at 80. Now, what is the efficiency of a pigment if we take up the oxygen partial pressure? So if you give more and more of oxygen to hemoglobin, what is the efficiency of hemoglobin? That it will take up all the oxygen molecules, it will transport, and it will release those oxygen molecules at the tissue level. So this is the efficiency of a pigment. But the second aspect of efficiency is that if a pigment can saturate itself at a low partial pressures, that pigment is more efficient than the other. So here you can very well see that the pigment of the elephant, the hemoglobin of the elephant, is the first one to be saturated to 98% at the lowest oxygen partial pressure, which is near to 55 or 56. If you see the first line, it ends up near to 98% percentage saturation, and this 98% saturation, it is achieved at near about like 50 or 55 mmHg of oxygen. So this is a very efficient hemoglobin in case of elephants. Second efficient pigment is in horse. Man comes at the third place. So the hemoglobin of man is good enough, but that is not better than the elephant or horse. So this is a comparison of the hemoglobins of different animals in the efficiency of taking up oxygen at different partial pressures of oxygen. So if you see one thing which is denoted by this blue arrow, that this arrow is at 100% HB saturation. So here 100% of the hemoglobin will be saturated with oxygen. Now you can very well see that none of the curves, they are touching the 100% level. So in nature there is nothing which is perfect. So nothing can be considered as perfect. So hemoglobin is also not going for 100% saturation at any oxygen partial pressures. Even at 120, you can see there is no curve which is touching the 100% saturation levels. So the level of percent saturation, it remains near to 98 in all the animals. Some of the animals like if you see the curve number 2 for horse, if you see the curve number 5 for fox, these are not going above 85 or 90. I should say 95. So this is again one factor which is affecting the efficiency of a pigment, and how, to what extent that pigment can be saturated if you have lots of oxygen molecules. So this is again a efficiency pattern or a parameter. So this particular graphs give you efficiency parameters of the hemoglobins in different animals. Now, this graph, it shows the oxygen dissociation curves in some birds. So, if you see the first graph, the upper graph A, this is for the bird Clara. You can very well, I think, see. This is for bird Clara. Now here, three graphs are given. One is black, second is red, third is green. Now the black curve is at the pH 7.5. The red curve is at pH 7.4 and the green curve is for pH 7.3. So that means if you decrease the pH, if you go acidic in the bird Clara, if you decrease the pH of the blood, what is the result? That Hb saturation will be decreased. Black, the first graph, more efficient Hb, it is going up, and it is going up very quickly than the red one. So that means the Hb saturation is achieved at like in lower times and up to higher levels at pH 7.5. So if pH is decreased at red one, the Hb saturation decreases. At the green one, it again decreases if Hb is further decreased to 7.3. So that clearly suggests that the decrease in the pH can decrease the percentage hemoglobin saturation in the bird Clara. Now, if you take the second graph, the B graph, you can again see three curves. Here you can see these are the three curves for three different birds. In the upper graph, we have got three curves for a single bird Clara. But here in the second graph, we have got three curves for three different birds. First is the green graph for bar-headed goose. Second is the black graph for emperor penguin. Third is the red graph for domestic duck. So this is a comparative chart for the oxygen saturation of hemoglobin at different partial pressures of oxygen. So here we can see that the hemoglobins of the bar-headed goose, these are more efficient to take up the oxygen. Second graph is for black. Penguin hemoglobin is again good as per the taking up of oxygen or the Hb saturation is concerned. And if we take up the third graph for domestic duck, the red one, this is lower in efficiency than the first two graphs. So it is, first of all, it is very slow in taking up the oxygen. Suppose if we take up the partial pressure of oxygen as 40 or 30, you can take 30 mmHg of partial pressure of oxygen. It is only saturated up to like 20%. It is quite slow. If we take the first two graphs, they are 20% saturated at 20 mmHg of PO2. So that's why we can very clearly see that the first two graphs are more efficient in taking up the oxygen than the third graph for the domestic duck. So here we can conclude that the hemoglobin of the domestic duck is lower in efficiency than the first two birds, the bar-headed goose and the emperor penguin. So this is a comparative chart, and you can give this chart in your examination if you want. Now this particular graph is showing the oxygen dissociation curves of the hemoglobin in different animals. Now here what we have taken is the black curve is for humans and the green curve is for small mammals or birds. So actually we cannot give a single line or a single graph line for all the mammals or small mammals or birds. But this green line is just given as an average of all the small mammals and birds. So it is a representative line for all the small mammals and birds. So it is compared to human. So human hemoglobin is much more efficient than the average graph line of small mammals. So here we can see that as the partial pressure of oxygen rises towards the right side, more of the hemoglobin is saturated for humans than the small mammals. So that clearly shows the efficiency of the human hemoglobin, that is better than the other mammals, small mammals or birds. Now this is a particular example of oxygen dissociation curve in llama. So as we know that llamas are found in mountainous environments. They are living on mountains, and on mountains we know that the partial pressure of oxygen, it is low. So they have developed a hemoglobin, a kind of hemoglobin which is far efficient than the humans. So this graph is showing the comparison of the llama hemoglobin, which is shown in the blue line, and the human hemoglobin, which is shown in the red line. So here you can very well see that the llama hemoglobin, it is more efficient than the human hemoglobin in taking up the oxygen. It rapidly oxygenizes, it rapidly forms the oxyhemoglobin. As you can, if you compare at the uh, partial oxygen pressure at suppose 4, the human hemoglobin is saturated only 30%, whereas the llama's hemoglobin is saturated at 40% at 4 PO2. And if you go at higher levels, if you take at 6, the human hemoglobin is saturated 70%, whereas the llama hemoglobin is saturated at about 85% at 6 PO2. So that means the llama has developed its hemoglobin in such a way that it is more efficient as it is living on mountains with the low oxygen levels. So it can uptake more of the oxygen efficiently and can take it to the tissues of the llama and can deliver it in time. Now, this is a very important aspect in the physiology of the respiration. This is what we call as the Bohrs effect. So this is a very important part. So every student must go for this slide very cautiously. Now, what is Bohr's effect? If we see the position and the shape of the oxygen dissociation curve, it is influenced by the concentration of carbon dioxide. So as we have discussed in the earlier slides, that if we increase the the PCO2, the carbon dioxide partial pressure, there is lower HB saturations. So the efficiency of hemoglobin, it decreases if the partial pressure of carbon dioxide is increased. Now, the increase in the partial pressure of the carbon dioxide, it typically shifts the oxygen equilibrium curve to the right side. Now, if you see this particular curve, we have taken up here the right shift of the curve. What we are taking up here is the right shift. If you see the green arrow, this is showing the right shift of the curve. Now, the shifting of the curve, it depends upon some factors. Now, what are the factors which affect the curve and which tends the curve to move towards the right? First factor is the PCO2. So this is the partial pressure of the carbon dioxide. So as we have discussed earlier, and you can very well see in the graph also, if the PCO2 is high, so here you can see on the right side, on the very right side, it is written as high PCO2. So it tends the graph to move towards the right. So this is the very first factor. If you have a low PCO2, then it will be on the left side. So this is the first factor. This is the partial pressure of carbon dioxide. The second factor is the pH. So you can see in the graph, if pH is low, the line, it tends to move towards the right. So that means if you have low pH, there will be low oxygen saturation. That means oxygen will give off the oxygen. Oxyhemoglobin will be broken into hemoglobin and oxygen. Oxygen will be released. And third factor is the temperature. If temperature is increased as it is present at the tissue level, the line tends to move towards right. So these are the main three factors which are affecting the oxygen dissociation curve. So this is for humans. This curve is for humans. So this particular effect is called as the Bohrs effect. Now the right shift of the curve by increasing the PCO2 or lowered pH or higher temperatures is called as the Bohrs effect. So what is the significance of this Bohrs effect? The significance is that it helps in the release of the oxygen from the pigment. So this is a very obvious kind of a thing. If you have higher PCO2, if you have lower pH, and if you have a higher temperature, the pigment will release the oxygen. So these are the three factors. These are the three conditions which are prevalent at the tissue level. So if we talk about tissue level, we have high CO2, we have low pH, we have high temperatures. So what will happen at the tissue level? That the hemoglobin will release the oxygen. So this is the significance of Bohrs effect. So conclusively, first of all, I should say what is Bohrs effect? So this is the right shift of the oxygen dissociation curve due to three major factors. First is the PCO2, second is the pH, third is the temperature. So what is the significance? It is the oxygen release at the tissue level. So that was all about the Bohrs effect. Now, very similar to the Bohrs effect, we have got the next effect, which is called as the root effect. The root effect is very similar to the Bohrs effect, but here we only and only consider the effect of the pH. So if you see this graph, you can draw it in the examinations also. We have got the oxygen saturation at one side, the vertical axis. We have got the partial oxygen pressure on the horizontal axis. So what we have in the graph is that the violet line is at pH 8 and the red line is at pH 7. So what is shown in this graph, that if we decrease the pH, it will decrease the percent saturation of hemoglobin. So this is the root effect. So this is again a right shift. So root effect is also a right shift as the Bohr effect. So this is the right shift. So what are the factors? Factor is the pH. So we are not talking about the partial pressure of carbon dioxide. We are not taking up the temperature as a factor. So here we only take up the pH as a factor for this right shift. So the root effect will decrease the percentage saturation of hemoglobin. So at the tissue level, if pH is decreased, it will give off the gas. Now students, we are to the second pigment after hemoglobin. This is the myoglobin. So this is a very similar kind of a pigment which is present in the muscles of the animals. So if we go with some features of the myoglobin, so iron and oxygen binding protein which is found in the muscle is the myoglobin. So it is again having iron as a moiety. So this helps in a fast uptake of oxygen and release of oxygen at muscle level. So it is related to the muscles. That's why it is myoglobin. Myo refers to muscles and globin is the same protein which is formed in the hemoglobin. So it is again having iron, but it is functioning at the muscle tissue level. Now, second point is it is related to hemoglobin. So it is very similar in structure to the hemoglobin. It is found in the bloodstream, and when it is released, the following muscle injury happens. So what happens that it is also found in the bloodstream. So it is released following the muscle injury. If the muscle it is injured, it is released into the bloodstream. So it is abbreviated as MB. As hemoglobin is written as HB, it is written as MB. So it is a single chain globular protein of 153 or 154 amino acids containing a heme. That means it is iron containing the porphyrin ring. This is a non-protein part. That's why it is called as a prosthetic group in the center around which the remaining apoproteins they fold. So very similar to the hemoglobin, but this is a single chain. So it has got 8 alpha helices and a hydrophobic core that will be seen in the coming slides. So it has a molecular weight of 16700 daltons. So it is not that much as we have got in the hemoglobin, but it is lower than the hemoglobin. It is the first protein to have its three-dimensional structure revealed. So this is one of the factors. It has an instant binding tenacity. So that means it can give you a hyperbolic oxygen dissociation curve. That means it can rapidly take up the oxygen. So if we go for the oxygen dissociation curve of myoglobin, in comparison to hemoglobin, I think the myoglobin will take up more of the oxygen in less of the time. So that means myoglobin is higher in efficiency than hemoglobin to some extent. Now, there are high concentrations of myoglobin in the muscle cells, and it allows the organisms to hold their breaths longer. That means uh, our muscles have got good concentrations of myoglobin, and as the myoglobin, if it is good in efficiency, it will take the oxygen first, then hemoglobin. It will take it to the muscles. So that oxygen is in a kind of storage in the muscles. So you can, if you are stopping your breathing for some time, so that will not affect the muscles as such because the myoglobin is having the oxygen in the stored condition for some time. So high concentrations of myoglobin, it allows the organism to hold their breath for longer. So it can help in the hypoxia conditions also. Now the diving mammals such as whales and seals, they have muscles with particularly high myoglobin abundance. That means if you are a diving mammal, so you are not taking frequent inhalations, if you are not taking oxygen frequently, then you need more of the myoglobin in your muscles. So there will be less formation of lactic acid in your muscles. There will be a late formation of lactic acid muscle in the muscles. So that means you can survive for longer times in water while diving if you have more of the myoglobin in your muscles. Now students, this is molecular structure of the myoglobin. So this is a single chain. First of all, the globin is a single chain over here as shown in the blue color. It is having a same ring. It is having iron in the very center as we have seen in the hemoglobin. But structurally, myoglobin is different than hemoglobin. Now, this is the curve for the, the fractional saturation, or you can say the oxygen saturation with the partial pressure of oxygen. Now this is a comparison of myoglobin with hemoglobin. Now you can see two curves. First is of myoglobin, second is of hemoglobin. So you can very well see that myoglobin is far much efficient than hemoglobin as for the percentage saturation or the fractional saturation is concerned. So if I take up like partial pressure of oxygen at 20, you can very well see the hemoglobin is only saturated up to like 0.25 level. It is very less as compared to the myoglobin. Whereas at 20, the myoglobin is saturated more than 0.9%. So this is the difference in the fractional saturation of myoglobin and hemoglobin. So here we can see that myoglobin is far better than the hemoglobin. So students, this is a comparative graph, and this is again a comparison between the myoglobin and the hemoglobins of human maternal blood, the human fetal blood, and llama hemoglobin. So as we have discussed in the previous slide that llama hemoglobin is far better than the human hemoglobin, but here we have taken two human hemoglobins, but they are again better than the adult human hemoglobins. They are better than adult human hemoglobins. What we have taken is the green line is for human fetal hemoglobin. So one thing I want to mention here is that fetus, the hemoglobin in the fetus is very good. It is better than the adult hemoglobin. So the green line is better than human adult hemoglobin. It is the fetal HB. The fourth line, the violet line, is for the human maternal hemoglobin. When the fetus is developing inside a female, the blood which is given to the fetus, the hemoglobin of that blood, the maternal blood, is taken over here. So it is again in a good efficiency. So we have taken the llama hemoglobin also as the red curve, but here you can very well see that the myoglobin, it far reaches above than these three hemoglobins in the percentage saturation with oxygen. So we can say that myoglobin is highly efficient in taking up the oxygen at lower partial oxygen pressures. Next is the hemocyanin. So this is the third pigment that we are taking up in this particular video. Hemocyanin is the main non-heme respiratory pigment. So non-heme refers to that it is not having iron as a moiety. So as the name suggests, if you see hemocyanin, so hemo, as we have referred as to the iron, so I think this is a misnomerous kind of a nomenclature about the pigments. So hemocyanin should have, a layman should see that hemocyanin should have an iron molecule, but here we don't have any iron molecule, so it is a misnomerous kind of a pigment name. So it has got a wide occurrence. It is found in many of the invertebrates. So it is always present in the colloidal solution form. It is a copper bearing protein. So this clearly shows that it is not having iron, but it is having copper as the metallic part. So because of the presence of the copper, it is giving a blue color to the blood. So it is a blue colored pigment. It is found in the two main phyla of the invertebrates, that is the mollusks and the arthropods. But one thing is should be mentioned over here, that the hemocyanins of both the mollusks and the arthropods, they are different in structure and the oxygen saturation. So that will be taken up in the later slides. So if we go for the molecular weight in mollusks, it is about 50,000 kDa, kilodaltons, and for arthropods it is 74,000 kilodaltons. So that's why I was saying that these are different in structure. Now students, this is a particular molecular structure of the hemocyanin. So I think you can very well see the presence of two copper moieties in the very center. So they are written as Cu. Now this slide, it gives you an idea about the difference in the hemocyanins of arthropods and mollusks. As we have seen in the previous slides, the molecular weight of arthropod hemocyanin was higher than the molluscan hemocyanin. So you can very well compare the sizes of these hemocyanins. On the left side we have got the arthropod hemocyanin, on the right side we have got the molluscan hemocyanin. What is the difference? That if you take the arthropod hemocyanin, it is having three domains. it has been written in the diagram domain 1, domain 2 and domain 3. Whereas the molluscan hemocyanin has got two domains, alpha domain and beta domain. So this is the basic difference in the structure, that arthropods hemocyanin have got three domains, the molluscan hemocyanin has got two domains, alpha and beta, while the similarity is that they have got a single active site. Both hemocyanins, they have got single active site in the center of the domains. The arthropod hemocyanin has got a single active site inside three domains, whereas molluscan hemocyanin has got a single active site inside two alpha and beta domains. So what it is doing, that it is attaching to two O2s in this kind of a manner. So if you see the oxygenation mechanism, we have got two Cu in the deoxy form. In the very left figure, you can very well see the presence of two Cu moieties, and if it is oxygenated, both the copper parts, they are binding to O atoms in this form. So this is the oxy form. So they are taking up oxygen in this particular structural form. Now the hemocyanin, it does not occur in the blood cells, but it is found freely dissolved in the hemolymph. So we have discussed earlier that some of the pigments, they are not present in the corpuscles, but they are present in the plasma. So they are dissolved here in the hemolymph. As we say, as we give a name, hemolymph, to the blood of invertebrates. So here we are not writing it as blood, but here we are referring it to as hemolymph. Now it forms the major protein constituent of this fluid, and in concentrations up to 120 milligram per ml. So it has got a huge concentration in the hemolymph, the hemocyanin. Now this concentration is about 90 to 98%. So that means 90% of the protein in the hemolymph is the hemocyanin. That means it is profoundly present in the hemolymph of the invertebrates. Now this constituent proportion, it depends upon the species also. So if you have different species of a genus, they may have different concentrations of the hemocyanin in their hemolymph. That is also dependent upon the age of the animal and the season in which you are considering the concentration of hemocyanin. So in some particular season, when the oxygen concentration is low, you may have higher amounts of hemocyanin in the hemolymph. So it is dependent upon the species, the age and the season. Now it combines with the carbon dioxide also. And how it leaves, that it leaves the CO2 or the monoxide, CO, when it is in contact with the air and regain its characteristic color. So that means hemocyanin can also combine with carbon monoxide very rapidly, and it leaves the carbon monoxide when it comes in the contact with the air. If it is having CO2 or oxygen, it will leave the carbon monoxide and it will regain its characteristic color, the blue color. Now, the next pigment is the hemoerythrin. So this is a iron bearing protein. It is having iron moieties. The molecular weight is only 10,000 kDa. So it is far lower than the hemoglobin, but it is higher than the hemocyanin. So it is found in the coelomocyte corpuscles and it circulates with the coelomic fluid. So it is again present in some of the invertebrates. So as invertebrates may have the coelomocytes which are present in the coelomic fluid, so hemerythrin may be present in the coelomocytes. So it circulates in the coelomic fluid. Now, what is the color? It is purple when oxygenated. So that means when it combines with oxygen, it gives a purple color to the blood or the hemolymph or the coelomic fluid, we should say. When it is deoxygenated, it is colorless. So that's why if we take up the hemolymph of some or the coelomic fluid uh, of some invertebrate, the coelomic fluid is colorless. The coelomocytes which are having the hemorythrin, they may be purple in color if oxygenated, they may be colorless if deoxygenated. So they are found only in a few groups of animals. If we see the structure of hemerythrin, we are having, you can see very well the presence of iron moieties. If you see the oxygen binding capacity of hemerythrin, you can see very well that we have got this iron moiety. It is taking up oxygen and it changes its form from ferrous to ferric. That means in the very first form, if we take up the deoxyhemerythrin, it is in the form of ferrous. That means Fe2 plus. But when it is oxygenated, it comes to the ferric condition, Fe3 plus, and it takes up the oxygen. But this association is of again temporary type. It releases the oxygen and comes back to the ferrous form. If we take this molecular structural diagram of oxygenation, you can very well see that the left side we have got two iron moieties. On the right side, when the oxygen is combined, you can very well see that one Fe, it is taking up two O atoms in a cyclic form. So this is the oxygenated hemerythrin structure. So these are the oxygen atoms which are taken up by the iron moiety. So here one thing is to be mentioned that only one Fe will take up the O atoms, the oxygen atoms. So oxygenation is at one level. So if we compare the arthropods and the mollusks for hemerythrin, they are also different. As we have previously discussed that the components, they are different in arthropods and mollusks. So here also we have different hemerythrin in case of arthropods and mollusks also. So it has been found that there is a remarkable difference in the carbohydrate content and monosaccharide composition between the hemerothrins from arthropods and from mollusks. So they are different in two things. First is the carbohydrate content. Second is the monosaccharide composition. Now students, we have got the last pigment, which is the chlorocruorin. Now this is an oxygen binding heme protein. Heme refers to the iron. So it is seen in annelids, mainly in the marine polychaetes like Chetoteris or Nereis. You can have the chlorocruorin. Now the oxygen affinity is weaker than the most of the hemoglobins. So it has got a very low oxygen binding capacity. So it is a dichromatic compound and it appears green in the dilute solution and red in the concentrated solution. So this particular chlorocruorin, it appears green in the dilute solutions and it resembles the hemoglobin, as in it is again in red color when it is in concentrated solution. Now the structure is very similar to erythrocruorin, which is likewise very similar to the myoglobin. So it contains many 16 to 17 kilodalton myoglobin-like subunits. So it is very comparable to myoglobin in structure. Chlorocruorin, it carries an abnormal heme group structure. So why abnormal? Because it is not comparable to the structure of the myoglobin. So chlorocruorin, it is found free floating in the plasma and it is not contained within the RBCs. So that means it is found in the plasma only and not in the corpuscles. Now if we see the structure of chlorocruorin, it is having iron in the very center, very comparable to myoglobin. So with this we cover up the whole of the pigments which are present inside both the chordates and the non-chordates, or we can say vertebrates and invertebrates. So that was the whole lot of topic which is called as a respiratory pigment physiology in your syllabus, and you may get some good questions from this part in your examinations. So first question may be like discuss the structure of the hemoglobin and discuss its functions. Or you may have a big question like discuss respiratory pigments among invertebrates and vertebrates. So that means that you have to give a whole lot of comparative study about all the respiratory pigments. So you may have a question on a single pigment like give the structure and function of chlorocruorin, or you can have a question on hemerythrin. So that means you may have some short answer questions also like what is Bohrs effect, what is a root effect, what are the factors which are affecting the oxygen affinity of the pigments. So that can be one of the question. You can have a question that give the physiology of respiratory pigments at lung and tissue levels. So that may be one of the questions. You may get a question on discuss the oxygen dissociation curves for respiratory pigments. So that means that you have to compare all the ODCs for all the pigments. So you have to make graphs. So for this particular question, you should be very particular about the structures and about the oxygen dissociation curves among different animals. So students, that was all about the respiratory pigments. So I hope that you have understood the basic concepts of physiology among different respiratory pigments.